to resume on our theme of antibody multivalency. And the next speaker is Gary Nabel, the President and CEO of MoDex. Gary is a former director of the Vaccine Research Center at the NIH and CSO at Sanofi. And Gary's going to speak on tackling AIDS, COVID, and cancer with multi-specific antibodies. Gary.
Here we go.
I don't see that one.
Yeah. Okay. Oh, we'll figure something out here. Let's see. Okay.
Do you want me to just...
I'll start with this, and I think...
the pointer, how do we get the pointer to...
let's see if it works up there. No? It's all right, I'll do it just by verbal. Anyways, thank you, Dennis. It's a pleasure to be here. I want to thank you for your organizing the scientific program, and to our colleagues here at Fair Journey for a really special meeting and a very special environment. I kind of feel like a king of Portugal coming to this place. What I wanted to do in the time allotted was to really give you a flavor for what we're trying to do with multi-specific antibodies. I'm going to spend a little time talking about the antibody platform, a little bit about our company and the platform, then go into some case examples as they apply to HIV, COVID, and cancer, really at a high level with a couple of more detailed examples, and then talk to you a little bit about some of our approaches to manufacturing and clinical translation. A little bit about the company. We spun out our team together, and I worked with my colleague Elias Zerhouni to found MoDex in November of 2020. Elias was formerly the director of the National Institutes of Health and president of R&D at Sanofi when I was the CSO at Sanofi, and we also connected during our days at NIH. And we had started to develop these multi-specific antibodies within the company, but we saw ways in which the technology could be taken to another level. And so we brought a number of our colleagues on board. My wife, Betsy, has joined as a strategy officer. Several colleagues from Sanofi. I'd like to point out in particular John Mascola, who succeeded me as the director of the Vaccine Research Center at NIH, who is our chief scientific officer, and then a variety of individuals, particularly Ronnie Wei, CJ, and Ziyang Yang, who had worked in our breakthrough lab, both at NIH and at Sanofi. So it's a seasoned team. We merged with a public company named OPCO. So we're a wholly owned subsidiary of OPCO. That occurred last May. That company is headed by Phil Frost. And interestingly, Dennis mentioned Richard Lerner yesterday. Richard was actually on the board of this company and has worked closely with Phil over the years, and so there's a little circle of life here, a circle of antibody life in the orbit of the company. So what I'd like to do is to share a little bit with you about what we do with multispecifics. You've heard a lot already about what others are doing, and looking forward to hearing many more talks. There's a lot of innovation, a lot of I'd say, advanced thinking about multispecifics that we didn't have in the early days. And it's a fun field to be in, particularly at this time when translation is really more opportune than ever. You all know about the constant and variable regions of regular immunoglobulins. What we do with our MSTAR platform technology, I don't know, can I I don't know if you can't see my pointer, can you? And I can't seem to, maybe try this. All right, well, I'll hand wave. So what you see on the right is that we've managed to assemble our multi-specific antibodies so that we can actually stack VHVL regions one on top of another. We have done this in a way where we can actually make the heavy and light chains with a single open reading frame, so that instead of using, say, 4 different cDNAs to co-transfect and give rise to a tetravalent bispecific, we can actually do this by with 2, because the heavy and light chains are combined in 1. And the advantage of that from is several-fold. I think at the from the manufacturing point of view and the laboratory point of view, it eliminates the. Oh, thank you. Let's see if that works. Yeah, there we are. Thank you. So it eliminates the need, the amount of mispairing that you may see in vitro when you're co-transfecting different vectors. The heavy and light chains are stoichiometrically going to be matched. There's no light chain mispairing. All of the binding sites are functionally active. And importantly, we not only can make the protein easily with this type of genetic engineering, but by having a more simplified cassette composition, it actually facilitates gene delivery. So we can make these multispecifics either with mRNAs or with plasmid DNAs, or if we wanted to, we could do it with viral vectors. This shows you, I think, one of the reasons we were attracted to the technology. We can make use of antibodies that are already well known, validated in some cases in human trials, and we can put the variable and light chain regions together in different combinations. And this is very typically what we do in the early screening process. We'll look at both different types of antibodies and in various combinations. And as you will see here, we can actually make up to 6 different VHVL regions, although typically these days we are working with 4 valencies just to keep things a little bit more simple. And this sort of goes into the flexibility of the platform. We start with these self-complementary VHVL regions, and then we can either make them, make all of them from the same VHVLs, from the same monoclonal, in which case we've generated a monospecific tetravalent. You heard about the advantages of avidity, and this is one way to achieve that using this, this format. We can also put them together in different combinations. And so, for example, here we can make a bispecific antibody that's tetravalent, and interestingly, this is because it's a homodimer, it can be coded by a single mRNA or a single cDNA, and so it gives us a lot of flexibility in terms of the genetic changes. Then you can see we can make a trispecific that's tetravalent. We can make a true tetraspecific. We've even gone up to the level of hexaspecific. Gets a little trickier when you get to hexaspecific, but it can easily be done. And this allows us to screen large numbers of diverse candidates rapidly, exploits these specificities, and probe for different orientations and valencies, and then to make use of the gene delivery where it's appropriate. So now to go into some of the specific case examples, let's start with HIV. We've been working on HIV for many years. It preceded us coming to MoDEX, even preceded coming to Sanofi, but I'll highlight some of the work that brought us to where we are today. The medical need in HIV therapy is, it remains still, I think, quite compelling, despite the fact that there are some very highly effective small molecule antiretrovirals. There are limitations to these molecules. I think in particular even the long-acting integrase inhibitors, at best, we're getting maybe 2 months of treatment duration, certainly with cabotegravir. There are some that may be a little bit longer, but they have other side effects, and I think those are going to limit their utility, particularly for preventive applications. As you all know, the antiretrovirals show drug toxicity in many cases. We see drug resistance. I think particularly important to patients is their perception of the need to take these often. And I think the longer the intervals between different doses, the more patients prefer those modes of therapy, and that's borne out in a variety of different real-world examples. And of course, I think we all recognize, despite an amazing amount of work, certainly Dennis has devoted a good part of his professional life to this. I did as well, and so did John Mascola. We sadly do not have a vaccine at the moment. And antibodies, I think, look promising, but we're not there yet. And I think that this is where maybe the multispecifics can make a contribution. In 2017, we published a paper on the trispecific antibodies. This was using our old platform called the CODV, the dual crossover variable region platform, where essentially we could put 3 different VH regions on a single molecule. And what we found when we did this was we could make trispecific antibodies that had greater potency and coverage. If you look at breadth on this scale and you look at potency here, these were among the most potent and broad antibodies that we had ever seen. It basically would cover close to 98% of the strains we would test on them. When we looked at the structure of the trispecific, and when we looked at the targets on the viral glycoprotein, again, these really built on the backs of years of successful antibody development against HIV, particularly against these highly conserved sites on the viral spike that have shown more breadth and more potency than a variety of antibodies that were defined in the early days of the field. And again, I think a tribute to the advances in molecular cloning and neutralization potency, and really our understanding of how to attack this virus. In that paper, we show the structure of the trispecific, and it's pretty much as we would expect it to be. We had the VRC01 antibody, which is a CD4 binding site antibody, on one arm, and then we stacked the other 2, one directed to the membrane proximal region and one directed to the supersite, the V3 glycan to the V1, V2 apex region. These were oriented in a way where there was not any steric hindrance, and they could bind independently to these sites. Since that time, we've worked with a variety of different groups to advance these into human studies. The one that's come the farthest has been the studies that were done with the AIDS Clinical Trial Group, sponsored by the National Institutes of Health, the NIAID. With our colleagues there, we did a dose escalation in humans of starting with 1 mg per kg, 3, 10, going up to 30, and then repeat dosing in all of this. I won't have time to go into all of the data here, but this will likely be presented in the next CROI meeting. What I can tell you quickly is that these showed very acceptable half-lives, better half-lives than many monoclonal antibodies, greater than a month. Nothing of concern in terms of safety, including anti-drug antibodies, and so we're optimistic based on what we've seen here that this multi-specific platform has some running room in terms of future development. Another assay that we have come to appreciate for the HIV applications has been the ability to suppress viral replication in blood derived from HIV-infected donors. So we take different donors we would culture them in vitro and activate the cells in such a way to bring out the virus, to bring it out from latency. And what you'll see, this is pretty complicated here because it's 5 donors, but if you look at any one of these donors, what you'll see is that essentially you will, with the tri-specific antibody, you will not be able to rescue virus from latency. Every one of these, if you use the single monoclonal antibody, the virus can escape, but with the tri-specific, we're able to maintain suppression for as long as we can. This is important to us, too, because one of the potential applications for in vivo treatment is not only prevention of HIV, which we are planning to test, but also suppression of viremia in people who have been treated with antiretroviral drugs. And so if we can suppress that viremia and give them a drug holiday, or depending on what the biology shows us, if it can help attack the reservoir, we might be able to get better forms of treatment in human trials. What I've shown you in the first few slides are the experience with our first generation tri-specific antibody. We have since made a second generation tri-specific antibody, this also in the CODV format. We've done this by including arms from other antibodies that are more potent, or by engineering some of the same specificities to make them more potent, doing structure-based design. And you can see that with this second generation tri-specific, here are 4 different candidates, but you can see that all of them are inhibiting at less than 1 microgram per mil at an IC80 of with over 98% of the strains being tested compared to the first generation, and the median IC80s are down quite a bit. So we think that it's in the vicinity of maybe 10-fold more potent. And the reason that's important is that when it comes to dosing in vivo, this may allow us a longer interval between dosing, and also allow us to stay above the minimum dose of suppression in terms of preventing viral recurrence. Now, having said all that, despite how promising these 2 early prototypes are, we have also been working on making another prototype based on the MSTAR platform that I told you about. And the reason is really shown here on the left. What we do when we look at our different monoclonal cocktails is we do a heat map, and we look at what the efficacy of each individual antibody is against our panel of 200 viral strains. And so, for example, the CD4 binding sites are exceptionally broad against most strains. Some of the glycan sites have little gaps in terms of the strains that they will affect. What we'd like to see typically is we'd like to get cross coverage by all 3 antibodies against all the viruses. That doesn't happen, but what we then do is we can say, well, then what are the best 3 that would give us the, say, the best coverage of all 3 versus all 2. Blue shows essentially no reactivity. So you can see that with this combination of 3, we're doing pretty well. We're getting greater than 95% neutralization coverage. Actually, we're not quite at 95. We're about 90% with this. But if we could add a 4th specificity, we think that we could now expand this to get dual coverage for more than 95% of the strains. And this, again, would help us achieve the higher potency. And so we've now started to make the 4 antibody combining site, the tetravalent ones. We have some candidates. We think we should be able to move those to the clinic, start moving them later this year. 2 CD4 binding sites, the 1 V1, V2 antibody, the GP41 Emperor, and we're looking at different combinations of their avidity and potency and protection in animal models. But we're optimistic that this can move forward. And needless to say, the advantage of being able to put all 4 molecules in one, as opposed to making 4 different antibodies, all of which have their own pharmacokinetics, all of which you have to bear the increased cost of goods and the product risk, that this is really an approach that has some attraction to it if we can move it forward. And then the final point I wanted to leave you with on HIV is that in the assays where we look at viral suppression, we've noticed something that is quite interesting, which is that there are 2 different CD4 binding site antibodies, both of which are really, really good. They'll hit 90+% strains of the strains we encounter. But again, if you look across all donors, you'll find that in some cases, one of those antibodies fails. So in this case, in this donor, VRC01 doesn't suppress, N6 does. In this donor, N6 suppresses, VRC01 doesn't. If we now do the mixture of donor 1 and donor 2, you can see that, as you might expect, neither of the monoclonal antibodies alone do, but if you use the combination of the 2 together, you actually can prevent escape. So there's value in terms of the valency and the selection of your specificities in terms of suppressing virus and preventing escape. And that's certainly one of the parameters that we're going to be following. And we hope this may contribute to better control in vivo. Now, obviously, while this work was going on, there was a tremendous amount of interest and activity in the COVID field, primarily spurred by the success of antibodies in treating acute COVID, not to mention the vaccines. And this, despite ongoing evolution of the virus, the antibodies were also highly effective for passive immunization, and this obviously is incredibly important to vulnerable populations, particularly the immune suppressed and those with underlying pulmonary disease like asthma or oncology patients. On the other hand, the new Omicron viruses evolved so rapidly that they rendered almost every one of the commercially used antibodies ineffective. And so at the moment, there are currently no antibody products for COVID. I think there are going to be some coming, but again, single antibodies, I'll tell you why we're worried about bringing more single antibodies into the clinic. Anyways, I think this just summarizes the medical need in terms of high-risk patients, and we also, as I mentioned earlier, have the ability to make these antibodies long-acting using FcRn mutations, also have the ability to deliver them genetically as either RNA or DNA. And then the other thing I think we shouldn't forget, for those of us working in the antibody field, is there's a lot going on with long COVID, and I think long COVID is actually probably a very heterogeneous set of diseases, but at least one subset of patients with long COVID do have persistent and ongoing viremia at a lower level, but it's there if you look. And so potentially, at least for that subset, this is something that could be investigated. One thing we found with COVID that we didn't see with HIV, but I think it was very encouraging to us, is that we actually can get synergistic neutralization of our COVID, of our SARS-CoV-2 viruses and reporter genes when we put the molecules and the binding site specificities together in the same molecule. If you compare the potency of the 2 monoclonal antibodies mixed together versus putting them on the same molecule, we get about a 20-fold increase in the potency at equimolar concentrations. So this is another example of where you can get this avidity effect, but based on differential recognition of epitopes. And the other thing that's really been quite interesting, and we've seen pretty reproducibly, is that with Nancy Sullivan at the VRC, and I should point out that these studies, as well as the HIV studies that I just described, have been done in collaboration with our colleagues at the VRC. Nancy and her team were able to pseudotype the SARS-CoV-2 envelope onto VSV and then do repetitive stimulations and repetitive infections in vitro, and then look at how many cycles of repetitive infection it took to become resistant to the monoclonal antibodies. And what you can see here is that here are two monoclonal antibodies. This bebtelovimab was the licensed antibody. I believe it was the Lilly antibody that was the last one to fall. But you can see in this in vitro assay, within 1 or 2 cycles, the virus becomes quite rapidly resistant to the single monoclonal antibodies. When we put 3 antibodies together in solution, you get less of that, and it doesn't happen to the same extent. But every time we use a trispecific or, basically the optimized multispecifics, we never get any kind of breakthrough. These are levels that are essentially background levels. And this has been done with now a variety of different trispecifics, and every monoclonal we put through the system here, you'll generate resistance mutants to it. So I think that another potential advantage of this approach is that it may allow us to suppress and prevent viral escape. And that could be good both at an individual level and on a population level. So we think this is something that's very much worth looking at. Now, we have generated multispecifics. We have tetravalent trispecific antibodies. We have tetravalent bispecific antibodies. And in all cases, we're starting with antibodies that are already nanomolar or subnanomolar active. And you can see here that we now have these tetravalent trispecifics that are incredibly active. These are certainly subnanomolar against every strain we've looked at, and that actually includes even the most recently emerged variants, the EG5. And so another advantage of being able to have multiple arms on the antibody, particularly for an evolving virus, is that it allows you to develop a repertoire of specificities that can protect you against some of the evolution that might occur in future times. And so we're looking at ways to move this now into the clinic. And this just quickly shows you that when we test these in the Syrian hamster model, they work against some of the more recent viruses. They prevent weight loss. They prevent viremia in the lungs and actually in the nasal passages. And so they are working both in vitro assays and in the best in vivo models that we've been able to look at. So lastly, just a word on some of our efforts in cancer immunotherapy. What we're moving forward with now are tetraspecific antibodies, where 2 of the arms are directed to T cells, and the T cell signals involve activation through the T cell receptor using anti-CD3, but then also activating through CD28, which is a signal 2 for the T cell that prolongs survival. You'll all remember that if you just give a CD3 signal in the absence of a survival signal, a signal 1 without signal 2, it leads to long-term T cell activation-induced cell death. And so being able to prolong the survival with a second signal is advantageous. We also can do that at the same time that we can target at least 2 antigens on the tumor. So that's designed both to increase the specificity of the tumor recognition and also to mitigate some of the consequences of escape. Here is the lead candidate that we're moving forward. It's against targeting 2 antigens that are found on prostate, gastric, and breast cancer. You can see in the presence of T cells, very potent, picomolar killing of all 3 cell lines compared to our controls. This slide shows the advantage of having more than one specificity in terms of viral escape. For those of you following the CAR T-cell field, you'll recall that one of the frequent modes of escape for CD19 CAR T-cells is loss of CD19. And so, for example, if we have, in this case, our 2 tumor antigens, we get very good killing. If we lose one of them, we still get equivalent killing, and if we lose the other of them, we still get equivalent killing. With a null control, we get no killing. So it provides a backup both to deal with heterogeneity of the target within the tumor population, and that's a real thing in tumors in vivo, but also in the event of immune selection as a consequence of treatment. So lastly, just to say a word about manufacturing and clinical translation, the point I want to really stress on this slide is that with the STAR platform, we've now solved the structure of these domains using cryo-EM in collaboration with the VRC. We find that essentially the structures we're seeing in our tetraspecifics are literally superimposable over natural VHVL, and we think that bodes well in terms of avoiding anti-drug antibody responses, although that needs to be done in humans. We have all the FC functions that Dennis described yesterday and that you heard about earlier today, so we can make modifications in FC. They have favorable attributes. We're getting very good cell yields comparable to what you see with standard antibodies. This is our pipeline, and this is really just to tell you that we are moving these forward into the clinic. We will have a tetraspecific, the CD3, CD28 that we call the laser, hopefully in early next year, in the first part of the year. We are moving forward with a leukemia lymphoma laser. And then on the infectious disease front, we hope to have some prototypes of the new format of HIV, and we already have some very advanced prototypes for SARS-CoV-2 that are ready to move into the clinic. And finally, from our perspective, some of the advantages of the multi-specific platforms, there are certainly concerns about bringing new platforms into the clinic, but the advantages are considerable. In the case of cancer or viruses, it allows us to target different sites. We can get synergy between those bindings. It allows us to cover antigenic variations, minimize escape, and to remain active in the face of continuing viral evolution. We can also take advantage of all the good things antibodies do and bring those into the platform as well. So we're excited to bring these candidates forward, and we hope that they'll make a contribution. I should also say we're a small company. We have a limited number of people. Because we have a platform, we're very collaborative, both with large pharmas and with small pharmas and with academics. And so if you have ideas, we would be more than open to talking about them. I'd like to then highlight also the people responsible for guiding these programs, John in particular that I mentioned, Vijay Chalchani, our Chief Technology Officer, and this amazing team, Ronnie, CJ, and Young, who make things happen magically in the laboratory. And there are others. I mentioned the VRC with our collaborations. I mentioned the HIV work with Dennis, and both at Scripps and at IAVI, Mark Feinberg, Devin Sock, our teams here at MODEX. And with that, I'll stop, and we can go on with the program.